All right, micro students, we are continuing into our understanding of microbial growth. So the last couple lectures that were on exam one were focused on how we can encourage microbes to grow or culture them in certain situations, which is often what we want to do. But then the flip side of that coin is when we want to control their growth or inhibit their growth. And so that's really what this lecture is focused on, different chemicals and physical conditions that can inhibit the growth of microorganisms. And so that's what we're going to head into. These definitions are likely in your vocabulary already, but we want to make sure that you understand the often small differences that exist between them. And so I'm going to run through these real quick and highlight what the major differences are. So the first two terms that I want to bring to your attention is antisepsis and disinfection. So those are two processes where chemicals are used to reduce or control the number of microorganisms on a surface. But the really important distinction between them, if you look, is antisepsis is going to be on living tissue. So say for a patient prior to surgery, if you're scrubbing a surface or prior to an injection or drawing blood, that's antisepsis. Whereas non-living tissue is not the best term, but just an object. So whether it's a countertop or if we're talking about surgical instruments, those are going to be disinfection. So that's the primary difference of those. Is it living tissue and non-living tissue? So we're going to talk about specific examples, but realize the exact same chemical could be used for each of these. So if you took alcohol and cleaned a surface, that would be disinfection. But if you took alcohol and scrubbed a patient's skin, that would be antisepsis. So just realize the distinction there. It's where you're minimizing the number of microorganisms. Pasteurization is a term you are likely aware of because it appears on different food and beverage items that you purchase. And so pasteurization is using heat to reduce the number of microorganisms in that food or beverage. But it is not going to remove all the microorganisms typically, but it focuses on two important groups. So the pathogens, so that term again is a microbe that may cause disease within humans, so we want to get rid of those within a food item. But then also those microorganisms that can cause spoilage of food, for it to start to look and smell and taste bad. And so that's what some microorganisms can do. So pasteurization is going to use heat to reduce those specific microorganisms to help increase the shelf life. Another way that we can reduce microorganisms, or a method, is called sanitization. And so this too is going to reduce microorganisms to a certain public health standard. And so that's the key of sanitization. It's getting below a certain threshold that we have deemed safe. So whether that's water quality, or if that is in the food service industry, how many microbes can be present on a cutting board or something like that. So that's what sanitization is. I didn't make a point of it, but I do want to go back and just highlight this. In each of these examples of microbial control, notice we've never said all the microbes have been destroyed or we've gotten rid of them. So this is a reduction in number. This is a destruction of most. It doesn't say all. This is reducing. This is getting below a certain standard. And so the final definition here is that definition where all microorganisms are going to be destroyed or removed from an object or some solution. And so sterilization is going to be the most rigorous control method that you could use. And so because of that, you can't do it all the time, but there are some instances where sterilization is what we're trying to achieve. Going back up, I skipped over degerming, but degerming is going to be very much coupled to antisepsis. And so it's really just coupling two processes that are working in conjunction with one another. So if you think about what I've described about using alcohol, for example, or iodine on the skin, very rarely would you just pour that on and just let it sit there, or put it onto some like a say a paper towel or something like that, or a towel, and it just sits there. But that's coupled with a mechanical action of physically scrubbing. You could also take like a brush and scrub, and that could lift the microorganisms off, but rarely would we use that by itself as well. So just realize that these two, degerming is the physical scrubbing action, and then antisepsis is the action of the chemical that can have its own effect. But those are so infrequently not occurring by themselves, but rather they're working, again, in concert with one another to be more effective than either one individually. Aseptic is similar to antisepsis in terms of spelling, just realize it's different. So aseptic describes an environment in which you're trying to reduce contamination. So this is what we use in a microbiology lab. We call it aseptic technique. And again, this is procedures or environments to try to minimize contamination. So for example, the Petri plates that we have, we want to minimize how much time the lids are off. And when we do lift the lids, you try to keep them kind of hovered over the Petri plate itself to ensure, or at least reduce, the likelihood of contamination. So we can describe those as aseptic procedures. The last two things I want to point out here are these are going to be endings or suffixes that can be tacked onto words. So side and sidal are related to one another. And so you can see bactericide or fungicide, or you can say, well, that chemical is fungicidal, or that drug is fungicidal. And we can contrast that with stasis and static. So again, fungistatic or bacteriostatic. And so the primary difference between these is when you see the word sidal, it means that the action is going to be to destroy that particular microbe. So bactericides destroy bacteria, fungicides destroy fungus, etc. But when you have the word stasis or static, that's going to inhibit but not destroy. And so that's a really important distinction if you're talking about a drug. If we're talking about that drug is fungistatic, but that one's fungicidal, it tells you about how effective it's going to be in reducing that particular microbe. So this slide is, if we're talking about control of microbial growth, when we apply either a chemical like a disinfectant or an antiseptic, or we're applying some physical control like temperature, we really want to understand on the level of the cell what is being affected that is inhibiting growth. And so there are really kind of two primary targets. Each of these is kind of broken down, but you can think of these as structures that are on the outside. And so you have to think back to the structure of cells. And so all bacterial cells are going to have cell membranes, and most of them have cell walls as well. And they're located on the outside of the cell, and they're really important at maintaining the integrity of the cell. And if either one of those structures is disturbed, it's going to likely lead to the cell rupturing or bursting, and then the cellular contents leaking out. That is a bad outcome for the cell, and that's how some chemical and physical agents can work. But there are also molecules that are going to be further inside the cell that are important as well. So we can talk about proteins, and we can talk about nucleic acids. Nucleic acids, if you don't remember, are going to encompass these two types of molecules, both DNA and RNA. And so proteins, if we start with proteins first, one thing to remember about them is they have each individual protein has a unique and characteristic structure, and that structure is going to be very much tied to how they function within the cell. So you'll hear this term a lot in describing proteins, that structure determines function. And when we start applying certain chemicals or applying certain physical controls like heat, you can get a change in the shape of the protein or a change in the structure. That's what this word denature means. So denature means a change in shape And so just realizing that that change in shape or structure of a protein is also going to lead to a change in the function. And usually it's to the detriment of that protein, so it's not going to function as well. You can also have these chemicals or physical agents working to alter or even destroy nucleic acids. So again, those nucleic acids are really fundamental to the function of a cell. So DNA and RNA are needed to allow the cell to survive through different mechanisms that we'll get to later on. So right now, just realize that DNA and RNA are important, and if they are altered or destroyed, it can negatively affect the cell. Okay. So we're going to look at this later on, but just realize those are the major categories that all of these chemical and physical microbial control agents are going to be working through. But as we get to specifics, we can attribute each individual chemical to either one or more of these. And so we'll see that as we get to it later on in this lecture. Much of this slide hopefully is pretty intuitive, but I'll just run through it fairly quickly. If you're considering a disinfectant, say, because you need to clean a surgical suite, we'll say, there are certain things that you would want to take into account. Obviously, you don't want something to be super expensive. You want it to act relatively quickly, and as opposed to it having to sit on a surface for several hours or something. Stable so that you can keep it stored and pull it out when you need it. And then this is hard to achieve, but this is the goal. You want some chemical or some method that's going to control all microbes, so inhibit their growth, but then we want it to be harmless to humans, but then other things that might be in an environment like precious objects or even animals, like we have pets in the house or something like that. So you want it to be safe for them. So that can be hard to do, but that's kind of the goal. And then the next category is looking at factors that can affect, the term here is efficacy, and that's just describing how efficient or effective these control agents are. And so realize that each chemical or physical method isn't going to be equal across all microorganisms. So there's something, I jumped around a little bit here, sorry. Degree of susceptibility. That just means that not all microbes are equally susceptible to things. Some might be easier to get rid of than others. Realize that different surfaces can allow for different types or more rigorous control. So for example, if we compare disinfectant on a surface versus antiseptic on a living tissue, the how strong the agent is, you can use a stronger agent for disinfectant purposes because it's on, say, a kitchen countertop. You wouldn't necessarily use those same chemicals on a patient's skin. So you may have to kind of dial back the intensity of that chemical so that it's not going to cause harm to the human skin. And then also there's environmental conditions that can affect the effectiveness as well, or the efficacy. So this is looking at that susceptibility spectrum that I was talking about. And so most of these should look familiar to you. And so just realize down here, most resistant. So this is going to be hard to kill on this end, or hard to get rid of, but I'm just going to write hard to kill. And then obviously on this one where it says most susceptible, this is going to be the easiest up here. So this is the easiest to kill. And this bottom one here, so the hardest, should not be surprising because we've talked about endospores, I think now at least a couple times. And so endospores are very resistant structures. So they're resistant to chemicals, they're resistant to temperature, and so they are by definition hard to kill. So not surprisingly, they're on this most resistant end. Backing up one, Mycobacterium, they have that thing in their cell wall that makes them a little bit different. They were an exception to the cell wall rule because they have mycolic acid that's also fairly protective. So they're hard to, we said mycolic acid was kind of a waxy molecule, so it protects them from different chemicals, whether it's disinfectants or antiseptics. This should look familiar to you because we saw this on our parasites. So when we talked about protozoans, there were two categories of them. and the trophozoite was the active form, and the cysts were kind of the dormant or the inactive. And so because the cysts are inactive or dormant, they are designed in a sense to be more better able to survive in the environment. So say Giardia, for example, in bodies of water that have been contaminated by different animals, you can have cysts of Giardia, and then if you drink that water, those cysts can develop into trophozoites. So those cysts are going to be a little more resistant or a little harder to kill than those trophozoites. And then bacteria. There are two types of bacteria that we focus on, gram-negative. Remember, there's an additional structure, an extra membrane on these guys that can serve to keep things out. So it's like having a double fence around your property. It's because you have two membranes instead of just one, you have the cell and the outer membrane. It's harder for some of those chemicals to get kind of to infiltrate into the cell to cause damage, whereas a gram-positive bacteria doesn't have an outer membrane. It only has the cell membrane. And so those tend to be a little easier to disinfect or susceptibility. We haven't talked a lot about viruses yet, but realize they're kind of down on the susceptible end, but not all viruses are equal. Envelope viruses versus non-envelope viruses. These are also called naked viruses. We'll get into that, but just realize that viruses are a little more susceptible. And then fungi are about in the middle, roughly. So depending upon what you want to achieve, you have to realize and think about, well, what kind of germicide would I want to use? And so here is a good refresher. There's that ending again, side or sidal. Here we see exactly how it's being used, which means to destroy or to kill. And a germ is again going to be some microorganism that can cause disease. So unfortunately, there's several terms that we can use to describe disease-causing microbes. So germs is one that's used, pathogens is another. But a germicide, obviously, then is something that kills germs. And so we can put germicides into different categories depending upon how potent it is. So if we're talking about a high level of germicide, that's going to get rid of all of these things, even up through endospores. That's going to be hard to do. It might be more expensive. It might be more rigorous. It might take longer. And so you have to decide if that's indeed what you want to do or not. Sometimes that's warranted. Other times it might be too much for what's needed. Intermediate germicide is going to dial it back a little bit. You can see it's going to get up through fungal, protozoan, cysts, viruses, pathogenic bacteria. So it's going to kind of get up to this area. So you're going to be removing these two from the mix potentially. And then low is even going to be lessening of the types of microbes that are going to be affected. So just realize that not all microbes are equally affected by different chemical or physical methods. So we're going to run through the physical methods first. Many of these also probably are aware of. So temperature is an important one, desiccation's another, filtering's another, and then radiation is the fourth. So there are slides dedicated to each of these. Not too much time is spent on any of them. Temperature by far gets the most attention because we probably use that most frequently, both in our house, but then also we use it in a micro lab as well. So temperature is what we're going to start with. So as we mentioned, there are certain targets for these methods, and so when you start to increase the temperature, one thing that's going to be negatively affected is, again, protein and their structure. The term is denature, or here it's denaturation, but same thing, denaturing or changing the structure of proteins. High temperatures also are going to affect those outer structures, so the cell walls and the cell membranes, and then also they can disrupt nucleic acids. So all high temperatures are going to target all of those potential targets, and so that's why high temperatures can be so effective at reducing and killing microorganisms. because it's so effective, there are certain measures that we use. And so these are two measures. And in the name, it kind of tells you what's going on. So a thermal death point is going to be a temperature. And so this is, if we apply a temperature for 10 minutes, what is the lowest temperature that's going to kill all of the cells? And so the thermal death point is a temperature that gives you some information. The thermal death time, obviously this is going to be a time. So this is at some temperature, so you can choose temperatures. How long do we need to leave a liquid at this temperature to achieve? So here's that term again, sterilize. And here it says kill all cells. And so you're reaching a very specific level of control. We want to get rid of all microorganisms or sterilize something. So we have the thermal death point and the thermal death time. They're obviously kind of related to one another, but you're measuring different things. One, you're measuring what temperature do you need to, so keep the time the same, 10 minutes, what temperature do you need? The other one, you keep the temperature, and then you have to figure out what, at a temperature, what time do I need to kill something? When we talk about heat, we can also talk about, is there moisture on the heat or is it a dry heat? So those are two different things that have implications for how well they're going to work. And so we'll look at moist heat first. Moist heat is going to be better. So it's going to be more effective than dry. And that just has to do with allowing that heat to better penetrate the microorganisms so that you actually have the effect. So remember what these terms mean: disinfect, sanitize, sterilize. So moist heat can be used to do any of those things. So for example, if you steam your floors, you could be using heat to disinfect things. You probably aren't going to get rid of everything unless you're very rigorous with your steaming, or sanitization, reducing to a certain public health standard, or sterilize, which is complete removal or destruction of microorganisms. So again, we've already talked about what heat does. It can target those proteins and those outer structures, the cell membranes and the cell walls. And some examples of moist heat. So if you boil water prior to drinking, that's using moist heat. Autoclaving is something that we do in the micro lab a lot, and this is to sterilize different things. So it's a really important tool in a microbiology lab. Also, surgical instruments can be sterilized through autoclaving. We've already talked about pasteurization, and you probably have seen different types of pasteurization. So if you buy milk in the refrigeration section, it's going to go through a regular pasteurization process. But then you can also have cartons of milk or chocolate milk, or pick your favorite type of milk, whether it's almond milk or whatever. They can just be at room temperature until you open them. And that's because they've gone through what's called ultra high temperature sterilization. So it is a type of pasteurization, but it's, as the name indicates, it's going to be much hotter than a regular pasteurization would. And that allows you to keep things at more room temperature for extended periods of time prior to you using it. So again, boiling is going to be obviously something that's been used for long periods of time to control microorganisms. Boiling is going to be effective against most things, but it's going to be hard to get rid of endospores. So how long you boil something is going to be really important. But again, there are some things that can survive this method. So again, not all methods are going to be equal to getting rid of all microorganisms. Autoclaving is going to be, as I mentioned, a really important way to sterilize things. So it will even get rid of endospores. And so that is the highest level of control that we're talking about. And the way it achieves it is by going above the temperature that water boils. And so you can do that by increasing the pressure. And so it's not only increased temperature, so it's above boiling. Remember, 100 degrees is where water boils. But when you increase the pressure, you can allow that solution to not boil, but get up to that temperature and really ensure that you're getting rid of all the microorganisms that are present there. And then we talked about pasteurization that's used. And so again, it doesn't sterilize things with regular pasteurization, but the goal is to get rid of those that might cause spoilage, or those microbes that might be potentially pathogenic. And there are different types of pasteurization, but again, the one that I mentioned earlier, the ultra high, is going to get to a much higher temperature that will allow you to store something for much longer, and even not have to refrigerate it for an extended period of time. So you've probably seen some of those items in the grocery store. So there's that ultra high sterilization that I was just talking about. So dry heat, as we mentioned, is not as effective, but there is one that will get rid of all living things, and that is to incinerate something. So that would be classified as a dry heat. So that's going to destroy and burn, so completely oxidize all the living tissue. That obviously can't be used because it will likely destroy much of what you're trying to work with. So we can use that for metal instruments. So you guys have like little tea lights, those little candles that you can use to sterilize your metal instruments in. So there you are incinerating something. So it's temporarily sterilized. Since your kitchen isn't a sterile environment, things can recolonize it, but you've done a pretty good job of getting rid of the microbes. Another one that you're obviously familiar with is refrigeration. So this is the opposite end of the temperature scale. And so this is really not going to kill a lot of microorganisms, but rather it's going to be that term that we saw before, static or stasis. And so it's just going to inhibit the growth. And so bacteriostatic. There are some microbes that actually do better at lower temperatures. So those are called psychrophiles. We learned about them in the growth lecture. But what's going to be true is most pathogens won't be able to grow well at 4 degrees Celsius, which is about what refrigeration is. And then also those that might spoil food won't grow as well. So it can extend the shelf life of food. Freezing can kill microorganisms, but it doesn't necessarily. So you can freeze things and thaw things, but there can still be microbes there. So this has to do with, so when you do thaw meat from a freezer, there still could be living microbes in it. So don't just assume that freezing process and thawing is going to kill them. They can still be viable in there. Desiccation is just the removal of water from a cell. So you're basically just drying out the cells. And so this was a really important way that people preserved food historically. It's still used today in some ways in some parts of the world, if you don't have access to power where you can refrigerate things. So by drying out meat, creating like a jerky, for example, then you can store that at room temperature and have a much longer lifespan of it. So again, there's that term, static. So this isn't necessarily going to kill all the microorganisms, but it's going to inhibit their ability to grow, so you can again extend the shelf life of something. Filtration is a process where you have something that has these really small holes in it that this isn't a really good example because it looks like the bacteria can fit through that. And so what you want is some sort of membrane or a filter that has holes that are so small that the microorganisms won't fit through them. So the liquid will pass through, but no microorganisms will. And so what you get at the end is a sterile solution. So we use this in the microbiology lab every so often to make certain solutions. So filtration and autoclaving are kind of the two primary ways that we get sterile liquids in our microbiology lab. Radiation is another way to reduce the number of microbes, because radiation is going to target a specific type And so radiation is going to negatively affect DNA in a cell. And so this can be used to irradiate food or to irradiate just different objects that we can then get rid of the microbes on. So like the Petri plates that we delivered to you guys were irradiated prior to putting them in those sleeves. And so then, or actually they're put in the sleeves and then irradiated. But nonetheless, you can have this radiation that's going to get rid of the microorganisms. So the key thing here is radiation is going to disrupt a specific target, which is DNA. There are different types of radiation and it has different effects on the DNA, but what we're mostly interested in is that radiation targets DNA. Okay, so those first several items were focused on physical control, so temperature or radiation, etc. So now we're moving into chemicals. And chemicals are going to fall into these 9 different categories. Some of these might be familiar, others may not be. And so again, the goal here is understanding the category. So do you know the different categories of chemicals? For each category, do you know what it's going to target at a cellular level? So does it target the cell membrane, or does it target the proteins, or target the DNA, etc.? And then some of the more common examples of each. And so that's what we're going to highlight as we move through this. So the first group are called phenols. So phenols or phenolics include things that you likely have heard of, like Lysol or Pine Sol. These are also what Lister used. So going back in your history lecture, there was that first surgeon who introduced using chemicals as antiseptics and disinfectants during surgery. So he used something that's related. It's called phenol, which we don't use today because it's a little more toxic. But there are still related compounds like phenolics that are used. And so as you can see, this is what they do. And then again, these are the examples. And triclosan you may be familiar with because it's put into some soaps, and so it's an antiseptic that is in some solutions that we can use. Okay, so that's what phenolics do. Alcohols is another category, and so alcohols include things like ethanol or isopropyl alcohol. This is also called rubbing alcohol. These are the percentages where they're going to be most effective. And so if you look at like hand sanitizers that often are alcohol based, they're not going to be 100% alcohol. It's hard to achieve anyways. But they're going to be some percent. So they do need to be a certain percentage to be effective. 60 is getting close, but 70 to 90 in most are going to be the most effective. This is what they are going to target as well. So they're going to denature. So there's that term again, denature proteins. And then also disrupt those cell membranes on the outside of cells. And then the examples are, again, the alcohols like ethanol and rubbing alcohol like isopropanol. Halogens is a term in chemistry that we use to describe things like chlorine and iodine. So they're a certain column within the periodic table. So we use chlorine to treat things. So if you have a pool, you likely have it chlorinated. We have chlorine in our water. We have fluorine in our water. There's iodine that can be used in surgical procedures. So there's a variety of different examples of halogens, but chlorine, fluorine, iodine, those are all examples. And this one is a little bit harder to pin down as to what specifically it's doing in a microorganism. And so this is the best guess. So this is pretty generic. It destabilizes cellular macromolecules. So that could be proteins, that could be something else, but we know it's effective, but we just don't have a great handling on the exact mechanism of it. So that one's a little harder to tease apart. Most people likely have peroxide at home, and it can be used as, depending on its percentage, it can be used to do different things. So if you buy like a brown bottle like this one, it's probably a 3% peroxide, and so that can be an antiseptic. You could potentially use it on surfaces as a disinfectant, but if you upped the concentration, you could even get to potentially the ability to sterilize. So peroxides, they're in their name, they are going to destabilize cellular molecules through kind of stealing electrons. They're called free radicals, but without getting into specifics. But oxidizing agents, which is what this belongs to, they're going to carry out oxidation reactions on things, and so they can start to break down and destroy other cellular molecules. We'll see this later in the semester, but we have already kind of talked a little bit about this. So when we talked about oxygen requirements of bacteria, there were different categories like strict aerobe and strict anaerobe. And so many organisms can and actually do need and use oxygen. And if they do, then they create these toxic forms of oxygen like peroxides and superoxides and hydroxyl radicals and things like that. And so many bacteria actually have enzymes like catalase that can neutralize some of these oxidizing agents. And so again, these may not be equally effective across all groups because some microbes have the ability to again neutralize or break down these oxidizing agents. Surfactants like soaps and detergents, so whether that is used to clean your clothes or to wash your hands, the term surfactant is it's going to act on the surface to help lift and dissolve things. And so they may not directly kill microbes, they can have that capacity, but they're really good at kind of binding them up in little kind of lipid balls, if is kind of the best explanation or the shortest explanation that I'll use. So it's going to help wash away those microbes, but it does have the ability to target, because these soaps and detergents have some lipid-like qualities to them, they can interact and disrupt those lipids that make up the cell membrane. So that would be the main target that's disrupted through soaps, or the bigger, more broad category would be called surfactants. If you buy Clorox wipes and things, there's some anti-septic solutions that have these quaternary ammonium compounds. They're called quats, or quat salts for short. So these are going to be harmless, at least to our knowledge, and so these are used extensively to try to disrupt the cell membranes of microorganisms, and they're used for either disinfectants or also antiseptics as well. And again, endospores are going to be at that hardest to kill end of the spectrum, and so depending on what we're talking about, this is not going to be as rigorous as some of the other methods, so it's going to be hard to, you won't be able to get rid of endospores with this type of chemical method. You may have seen this logo on things. So there are certain metals that we know that have antimicrobial properties. So things like copper and silver. And so they're starting to put those on certain surfaces to minimize the amount of microbial growth. So heavy metals, again, we see where they're going to affect is at the level of proteins, and that can inhibit the function of proteins and lead to, so again, notice static and static. So it's not going to kill them, but it's going to inhibit their function likely. So one example of this is silver nitrate that can be put in the eyes of offspring of kids delivered that may have been exposed to the gonorrhea bacterium. And then also antibiotics. This can kill other microbes too, potentially, that might cause damage to the developing eye. We see this in vaccines, so thimerosal, which is a heavy metal that can be used to help preserve vaccines so they have a longer shelf life. And then also, if you've ever had to buy like an algicide because you have algae growing in your pool, if you look at the chemical composition, frequently you'll see that it's got some copper in there and stuff like that. But okay, This, you probably have heard of formaldehyde and formalin. We use this frequently in fixing tissue or preserving tissue. So we see this if you've had to dissect organs or animals. In the past, this is what we use, but because it has such a powerful chemical effect, it's not super safe to use. So we've kind of moved away from this, but it has a certain mechanism of action. And so these aldehydes, like formaldehyde and glutaraldehyde, what it does is it's going to change the shape of proteins and then also change the shape of nucleic acids. So both of these two types of molecules are not going to be able to function. So that's how aldehydes are going to function. This last group, the name kind of tells you these are going to be gases. So these are certain gases, and they too, similar to the aldehydes that we just saw, are going to target those inner molecules like proteins and DNA. So these aren't going to be used extensively, but they are used in some medical fields. So they can be in hospitals and dentist office. But you have to have certain types of equipment for them to be used appropriately. But that's what these gaseous agents are. There's one more category. We're not going to get into a lot of them here, but one more category of chemicals that are used to control growth. And so these are used for therapeutic purposes. So all the previous chemicals were used to disinfect surfaces or potentially sterilize things, or maybe treat a surgical site prior to surgery. But the next lecture, so lecture 8, we're going to look at different categories of antimicrobials, including antibiotics, some antivirals, antifungals, etc., that we can use as well. And so they're in the same category as controlling of growth, but we want to give them their own lecture to better understand them, because they're really important in treating disease. So that will be the next lecture, though.